Good day everyone, my name is Lubabalo Mofu and I'll be giving a talk entitled Feeding Ecology of Co-occurring Fishes in the Sanders River Valley Irrigation Ponds Assessed using Stable Isotope and Gut Content Analysis. I am a postdoc at South African Institute for Aquatic Biodiversity and I'm currently working on biotic interactions between native and non-native fishes in novel environments. Freshwater ecosystems are amongst the most imperiled ecosystems in our planet as a result of the introduction of non-native species, habitat destruction, and biological invasion. Freshwater fishes are amongst the most introduced taxa through human actions, and the negative effects of introduced fish include biodiversity loss, biotic homogenization, and destabilization of native freshwater communities. This introduction of non-native species can have significant effects on native individuals, populations, and communities. And therefore, understanding how species potentially compete for the same resources is of great practical and conceptual importance. Quantifying interspecific interactions between native and non-native species within a particular ecosystem is integral to understanding community biology through provisioning of food web dynamics data on competition, resource partition, and predation. Stable isotope analysis is comparatively cost-effective and can provide information on the long-term assimilation of dietary food sources, as well as inform on the trophic ecology of predator-prey interactions. Stable isotope analysis is an important tool in the study of ecological food webs and has been used to complement gut content analysis. The carbon isotope signature provides information on the habitat use of predators, the sources of carbon from the bottom of the food web, and the overall diet preference of the predator, while the nitrogen isotope signature is used to calculate the trophic position and can be used to measure energy transfer. Gut content analysis is considered a standard practice for identifying food sources and can provide direct evidence of an organism's potential food resources. However, this method requires large sample sizes and is only a snapshot of a consumer's diet. To understand the effects of alien fish invasion through human activities, the Sanders River catchment in the Eastern Cape in South Africa provides an exceptional network of ponds constructed for irrigation purposes. The Sanders River irrigation scheme was completed in 1975 primarily to provide water from the Orange River to a network of dams. And since then, it has resulted in the passive introduction of fishes, and amongst those include Glossogobus calidus, Telcrystella estuaria, Oreochromis mozambicus, and Gambusia finis. Previous studies conducted on these ponds include a study by Woodford et al. In 2013, where they examined the fish composition upstream, within and downstream of the Sanders River Valley irrigation network. Their study showed that proper pressure from the irrigation network contributed to the successful establishment of Glossogobus calidus, Telcrystella estuaria, Oreochromis mozambicus, and Gambusia finis. Howard et al. In 2013 studied the population dynamics of Gambusia finis in the same irrigation ponds where Glossogobus calidus occurs, and they found that the abundance of Gambusia finis was not only driven by water temperature, but by also the presence of Glossogobus calidus and competition for resources. Glossogobus calidus is also known as the river goby. This fish is native to South Africa and feeds mainly on benthic resources. Telcrystella estuaria is also known as the estuarine round herring. This fish is also native to South Africa and feeds predominantly on zooplankton and phytoplankton. Oreochromis mozambicus is also known as Mozambique tilapia. This fish is also native to South Africa and is widely distributed and can feed on different kinds of prey such as algae, phytoplankton, and zooplankton. Gambusia finis is commonly known as western mosquito fish and is native to North America. Gambusia finis feeds mainly on algae, crustaceans, and insects. The aim of this study was to describe and compare the food web structure and trophic fish community characteristics of the Sanders River Valley irrigation ponds and assess the trophic interactions between Glossogobus calidus, Telcrystella estuaria, Oreochromis mozambicus, and Gambusia finis. Stable isotope and gut content analysis were used to test the following hypotheses. The invasive Gambusia fenis would exhibit non-specialized feeding habits and would show variation in dietary composition across size classes, and species consuming a more varied diet like Orycromus mozambicus and Gambusia fenis would overlap with Glossogobus callidus. Samples for stable isotope and gut content analysis were collected from 4 irrigation ponds in summer, and these ponds are located in the Sinus-Dua Valley in the Eastern Cape. The sampling period was chosen as it coincides with peak species abundances. Fish were collected using a seine net. Phytoplankton and zooplankton were collected using a plankton net. Microinvertebrates were collected using a SAS net. Plants and detritus were collected by hand along the lateral zones of the 4 irrigation ponds. Upon collection, all samples were identified to the lowest possible taxon. Fish samples were taken for total length to the nearest millimeter, and the demersal tissue was taken for stable isotope analysis. Once sorted and identified, samples were oven dried at 6 degrees for at least 48 hours, then ground into a homogeneous powder and sent for isotopic analysis. Standard ellipse areas were calculated to measure the trophic niche width. The Bayesian stable isotope Bigman models were used to assess the relative proportion of each food source in the diet of the 4 fish species. To assess the diet composition of the 4 studied species, the gut contents of Glossogobus callidus, Stella estuaria, Orycromus mozambicus, and Gambusia fenis were examined. The total length of each fish was measured to the nearest millimeter and then dissected. For each prey taxon, the index of relative importance was then expressed as a percentage of the index of relative importance of all prey categories. Stable isotope analysis showed that the niche space occupied by Gambusia fenis is broad and overlapped with that of the other 2 species. In addition, Gambusia fenis and Orycromus mozambicus have the largest isotopic niche compared to Glossogobus callidus and Stella estuaria. The isotope matrix shows that Gambusia fenis and Orycromus mozambicus utilize a wide range of resources and trophic levels. The isotope niche of small-sized fish showed a variable niche overlap. However, despite high degree of overlap observed, subtle differences were observed for large-sized classes. Stable isotope results showed that Glossogobus callidus is a generalist insectivore and feeds predominantly on benthic resources. Piscivorous feeding behavior with increasing body size was also observed. Glossogobus callidus had a narrow niche breadth and occupies a higher trophic level. Stella estuaria is a generalist feeder capable of exploiting any resources. This fish feeds on phytoplankton. This species had a narrow niche breadth and occupied a higher trophic level. Orycromus mozambicus is an omnivorous feeder, whereby juvenile feeds on phytoplankton, while adults feed on phytoplankton and zooplankton. This species utilized a wide range of resources, fed across multiple trophic levels, and at large isotopic niche width. The results obtained from stable isotope analysis suggest that Gambusia fenis had a broad dietary niche, feeds on a wide range of resources, feeds across multiple trophic levels, and at large isotopic niche width. The gut contents of both small and large-sized Glossogobus callidus contain mostly insects and detritus. The gut contents of both small and large-sized Stella estuaria contain cyanobacteria and rotifers. The gut contents of small-sized Orycromus mozambicus contain mostly detritus and algae, while the gut contents of large-sized Orycromus mozambicus contain detritus and rotifers. The gut contents of small-sized Gambusia fenis contain copepods, while the gut contents of large-sized Gambusia fenis contain copepods and insects. Gut content analysis shows that Glossogobus callidus is a generalist insectivore. Detritus forms bulk component of Glossogobus callidus diet. This suggests that high contribution of detritus may be accidentally ingested, most likely during benthic foraging for invertebrates. Overall, gut content analysis results of this study demonstrated that Gilchristella estuaria is a generative feeder, and to date, only limited literature exists on resource use by Gilchristella estuaria, and current literature is based on estuaries, where Gilchristella estuaria feeds predominantly on zooplankton, including copepod eggs. The gut content found in this study agrees with findings by Babla et al. In 1981 that the diet of Gilchristella estuaria is mainly comprised of zooplankton. The gut contents found in this study agrees with Costello et al. In 2015, who demonstrated that in turbid waters, Gilchristella estuaria adapted their feeding strategy to a non-filter feeding strategy. Results from gut content analysis suggest that Orechromos mozambicus is an omnivorous feeder, whereby juveniles feed predominantly on insects and phytoplankton, and adults feed on insects, phytoplankton, and zooplankton. The gut content analysis suggests that Gambusia finis had broad dietary niche. This is similar to other research on this species, which has shown that Gambusia finis feeds on a wide range of resources, including algae, detritus, zooplankton, and crustaceans. Small-sized Gambusia finis feed predominantly on copepods, while large-sized Gambusia finis fed mainly on copepods and insects. This suggests that large-sized Gambusia finis had greater efficiency in predation and ability to move up and down the water column. Results from stable isotope analysis demonstrated considerable niche overlap. Gambusia finis had broad niche space. Gambusia finis and Orechromos mozambicus had the largest isotopic niche width. And in addition, stable isotope analysis suggested interactions between Glossogobus calidus, Orechromos mozambicus, and Gambusia finis. Gut content analysis suggested well-partitioned feeding niche. Glossogobus calidus, Orechromos mozambicus, Gambusia finis fed predominantly on benthic resources, while Gilchristella estuaria fed mainly on plankton resources. And in addition, gut content analysis suggested interactions between Glossogobus calidus and Gambusia finis. With regard to our first hypothesis, stable isotope and gut content analysis showed variation in the diet of Gambusia finis across size classes. Thus, we accept the first hypothesis. For the second hypothesis, this study highlights the simplicity of the food web within the Sanders River Valley, where food items are shared by all small-bodied species. Stable isotope analysis suggested interactions between the littoral groups such as Glossogobus calidus, Orechromos mozambicus, and Gambusia finis. Thus, we accept the second hypothesis. Given that during summer period there are high abundance of food resources, this could suggest increased interactions between Glossogobus calidus and Gambusia finis, which both fed on insects. Overall, the wide dietary niche of Gambusia finis could have facilitated the success of the species within these irrigation ponds. Stable isotope and gut content analysis provided greater ecological insights that are specific to the invasive Gambusia finis. Quantifying the impacts of invasive species can be challenging due to complexity of ecological interactions, particularly in aquatic environments. Stable isotope analysis has been shown to be a useful tool for tracking changes in trophic structure and provide a powerful approach to predicting the invasion impacts of non-indigenous species. I would like to thank the following people. My name is Lubabalo Mofu, and thank you very much for listening.